Benu, thank you very much for joining us for this special research rounds here at the Heart Institute and also in the region as well. And so before we introduce our honored guest, and just want to make sure in terms of the logistics in that, thanks for folks who are also joining us on virtual connections in addition to those of the folks who are in the room here. Really appreciate it. And so keep in mind, if you're actually on the virtual connect, that if you have any questions, please use the Q&A feature and during the progress of the lecture itself, and we'll answer those questions and give Eric a hard time after his presentation. But for those who are interested in posing questions directly in the room, please use the microphone, and so that you will be identified also on the virtual broadcast as well. And if you have any technical issues, particularly if you're having difficulty connecting, please use the chat feature, and the team will make sure that you're able to connect successfully. And so we are absolutely delighted today to have Professor Eric Saronen to give us a presentation of his research and up-to-date insight into how biomaterials and also some of the toxic byproducts from myocardial infarction can be a challenge, but also can be tamed. And he's a professor of surgery at University of Ottawa, and also cross-appointed to the Department of Cellular Molecular Medicine, in fact, managing some of the top programs within the CMM. And he's also a member of the Division of Cardiac Surgery here at the University of Ottawa Heart Institute, and of course, the very well-known BEATS research program. And you see the logo there, which is really well-known through some of the communications. And so this is a fantastic brand that we continue to promote as well. And he did his biology degree as an undergraduate in 1996 and a PhD in cellular molecular medicine in 2004, and subsequently postdoctoral fellowship at University of Ottawa Heart Institute. And with this, he has actually developed a significant program in terms of understanding the interaction of biomaterials, cardiac regeneration, and defense against cardiac injury, and as well as very important insight into some of the biochemical processes following cardiac injury and some of the impact of this in terms of adverse remodeling, and which you will hear some from, particularly in terms of methylglycol, and also ways to potentially tame this, both biochemically as well as using biomaterials. And his work is published in high-impact journals, and Eric has actually received the Researcher of the Year award, as well as Dr. Robert Roberts Award, for outstanding publications several years in a row. And he received his funding support from CIHR, Heart and Stroke Foundation, NSERC, Diabetes Canada, and many others. And truly is a leader, in this unique field, and provides translational model work, really, in terms of the surgical arena, in terms of understanding the biological basis of disease processes and finding unique solutions. So it's really actually a delight today. We look forward to Dr. Saronen's presentation. It's entitled Dealing with the Methylglycol After Myocardial Infarction. Eric.
Thank you, Peter, for the kind introduction, and thank you all for being here. So today I'm going to talk about methylglyoxal after myocardial infarction and how it can be targeted therapeutically. So I thought I would present a lot of unpublished data today to give you an update on the projects that we currently have ongoing in the lab. But first, before getting to that, I'll explain to you what is methylglyoxal and what is the research that we've done that have led to our current projects. Slides won't advance. Oh, there we go. Okay, so methylglyoxal is a highly reactive dicarbonyl compound, and it's produced primarily as a byproduct of glycolysis. There are other sources, but 90% of it is derived through the glycolytic process. Now, it's a concern biologically because it is a primary precursor in the formation of AGEs, or advanced glycation end products. These AGEs are proteins or lipids that become glycated due to exposure to free sugars and reactive carbonyls. So here are several types of AGEs that are formed by methylglyoxal. The primary adduct formed by methylglyoxal is MGH1, which is shown here, and I will be talking more about this throughout the lecture. Now, glycation of proteins can lead to protein and cell dysfunction. So the advanced glycation products, end products, are formed inside the cell, and so they can damage the cell inside. But in addition to this, MGHs in the cell can be released into the environment and target AGE receptors, and then this targeting of AGE receptors then triggers adverse effects in neighboring cells. So it's a multiplying effect when you have the generation of these methylglyoxal and its AGEs. This triggering of the AGE receptor then leads to further reactive oxygen species production, inflammation, and oxidative stress. Now, the body does have an enzyme system that's capable of dealing with methylglyoxal production. So in a healthy individual, the glyoxylase system, which has two glyoxylase enzymes, glyoxylase 1 and glyoxylase 2, it is able to convert the methylglyoxal that has formed along with the cofactor glutathione into lactate, which then is excreted from the body. However, conditions such as hyperglycemia, inflammation, oxidative stress, hypoxia, and ischemia can overwhelm the glyoxylase system, leading to the accumulation of methylglyoxal. The accumulation of methylglyoxal then has been shown to play a role in the pathogenesis of numerous diseases, including a lot of metabolic disorders, and in particular, it has been studied in diabetes due to the glycolytic pathway. However, not as much is known about the role of methylglyoxal in cardiovascular disease, and in particular, myocardial infarction. So years ago, we set out to look at possible effects of methylglyoxal post-myocardial infarction and published this study in 2017. This work was led by a PhD student in the lab at the time, Nick Blackburn. And I'll go over some of the main findings of this work, which laid the foundation for much of the work that we're doing today. So we used a transgenic mouse in which glyoxylase was overexpressed in the endothelium and in the bone marrow. And then just to confirm the model as shown here, when you look at the whole heart, glyoxylase 1 expression is increased in the transgenic mouse. In the endothelial cells obtained from the heart, you have significantly greater expression of glyoxylase 1 enzyme, whereas cardiomyocytes, which did not express glyoxylase 1, was also confirmed as shown here. So these transgenic mice were then compared to normal control mice. MI was induced surgically, and then we isolated tissue at 6 hours and at 4 weeks and followed these mice up over this period of time, performing echocardiogram to look at function and immunohistochemistry, as well as some other molecular assays looking at methylglyoxal production. So here you can see that in the that MGH1, which is, as I mentioned before, the main AGE that is formed by methylglyoxal, is increased in wild-type mice, even just 6 hours after myocardial infarction has been induced, whereas the mice that overexpressed glyoxylase 1 were protected from this increase in methylglyoxal age formation. And this was maintained, so the cardiac function then also was shown to be improved in the mice that overexpressed glyoxylase 1 at 7 days, and this was maintained out to 4 weeks. In addition, GLO1 overexpression reduced the scar size. So in summary then, the levels of MGH1 or MGAges increase in the heart at 6 hours, already at 6 hours post-MI, and overexpressing glyoxylase 1 in the vasculature was able to prevent MGH1 accumulation. We also looked at the effect of overexpressing glyoxylase 1 on cell death and vascularization. As you can see here, the number of cardiomyocytes that were dying in the transgenic mice was reduced, and also the total number of cells in the myocardium that were dying was reduced with the GLO1 overexpression. We saw increased vascularity in terms of both the numbers of capillaries and arterioles in the animals that overexpressed glyoxylase 1. And we also looked at the targeting of glycation specifically in the collagen. Collagen is a primary target for methylglyoxal due to its long half-life and its slow turnover. And it also has several amino acid residues that are targets for methylglyoxal. And as you can see in the wild type, there is a significant overlap of the MGH expression and collagen, which is the yellow-orange overlap here. And there was much less of this observed in the mice that overexpressed glyoxylase 1. So not only are the cells being protected, but the extracellular matrix in the hearts of these overexpressing mice is also protected. So in summary, methylglyoxal and its ages, specifically MGH1, contribute to adverse remodeling, reduced vascularization, and impaired cardiac function post-MI. Collagen in the myocardium is a target for glycation by methylglyoxal, and overexpression of glyoxylase 1 can reduce the effects of methylglyoxal and improve cardiac function. So this study identified a causative role for methylglyoxal in the deterioration of cardiac function post-MI, and thus constitutes a possible target for therapy. So these are the studies that I'm going to be presenting today that are ongoing in the lab, or yes, one of them is published, and I'll be showing some of those results as well as what we are currently doing on that project. So we'll be showing you studies with our collagen hydrogel therapy, which you are most of you I'm sure are aware that Mark, Emilio, and I have been working on for years, and only in the last few years discovered a primary mechanism through which this confers beneficial effects is through its ability to deal with methylglyoxal, and I will be showing you that. Also be showing you research using GLO1 loaded nanoparticles for reducing methylglyoxal effects. Also explain how exercise training may be conferring some of its benefits through reduction of methylglyoxal ages. And I'll be going over some of the new research that we've started in the last year, looking at the role of methylglyoxal in the brain-heart connection. Of the main findings of our human recombinant collagen hydrogel work that was published in 2019. As you can see here in the graph, cardiac function in terms of left ventricular ejection fraction was improved when we treated mice at 7 days post-MI using our collagen type 1 material. This was the only material compared to collagen type 3 that resulted in improved function, whereas type 3 simply maintained cardiac function. The type 1 material was also able to increase vascularization in the myocardial tissue, recruit a greater number of wound healing macrophages, reduce scar size as well. And so, as I explained earlier, collagen is a primary target for methylglyoxal. And since our material that we've been working with, it is consists primarily of collagen, then perhaps it is working as a sponge to sequester methylglyoxal from the post-MI environment. And this is work that was investigated by a PhD student who's since graduated, Sarah McLaughlin. And I'll go over the published results of her work. So again, mice underwent surgery to induce myocardial infarction. They were then injected at 7 days post-MI with the collagen hydrogel. And then we isolated tissue at various time points, as well as looked at cardiac function through echocardiography. As you can see here, already at 2 days post-MI, the collagen matrix treatment improved cardiac function in terms of ejection fraction compared to PBS control and also compared to baseline. We also observed improved contractility in the heart already at 2 days post-MI, which was maintained out to 28 days. So this is demonstrating that the collagen material is having an immediate impact in improved cardiac function after its delivery 7 days post-MI. Since we were expecting that it would be possibly targeted by methylglyoxal, and methylglyoxal is a potent inducer of oxidation, we looked at oxidative stress in the tissue after treatment. So we looked at 4-H&E, which is an oxidation product. And as you can see, it was significantly reduced with the collagen treatment, collagen hydrogel treatment in the remote myocardium and when we looked at the total left ventricular tissue. We also saw that the biomaterial treatment was reducing cell death. And we saw this in the border zone and the remote myocardium of the hydrogel treated mice compared to PBS treatment. And we saw that specifically also in reduction in the number of tunnel positive cardiomyocytes. So it's protecting all of the cells, including cardiomyocytes, just even 2 days after the hydrogel has been delivered. So looking specifically at its ability to reduce MGHs, we did some immunohistochemistry and observed that MGH1, so again, the main age product of methylglyoxal, is reduced in the myocardium of hydrogel-treated mice compared to PBS. And there's reduced MGH1 observed accumulating in cardiomyocytes, as shown here. And this was seen in all 3 areas in the infarct, border zone, and remote myocardium. So it is having a protective effect on the cells, likely through limiting the accumulation of methylglyoxal and its ages. So looking specifically at the ability of the material that we injected to mop up the methylglyoxal, we fluorescently labeled the injected matrix with a red fluorescent dye, which you can see here. And then we counterstained with an antibody for methylglyoxal derived H1 ages. And we saw co-localization of MGH1 and collagen in 52% of the injected material, whereas 48% was unmodified. And again, this is just 2 days after it has been delivered into the myocardium. So in summary, upon injection, the collagen hydrogel acts like a sponge to soak up methylglyoxal from the post-MI myocardial environment. The removal of methylglyoxal is associated with a reduction in oxidative stress and increased cardiomyocyte survival. So removing methylglyoxal from the myocardial environment by providing an alternate glycation target, which is the collagen hydrogel, can reduce oxidative stress and promote cell survival post-MI. So continuing from that work, just to further confirm that it is the ability of the material to soak up methylglyoxal that is contributing to the beneficial effects, we have a current PhD student in the lab, Gigi, is pre-glycating the material that we inject into the heart. So if the target sites are already glycated, then it should not be able to remove the glycated the agents from the myocardial environment, and thus we would expect reduced cardiac an inability to protect the myocardium. So as in the previous work, we've delivered now this pre-glycated matrix at 7 days post-MI. And so this is some of the preliminary data we've obtained so far. You can see here that, again, the hydrogel itself is able to improve cardiac function in terms of the ejection fraction 28 days post-delivery, whereas the pre-glycated hydrogel is not able to confer this functional effect. And we similarly see effects on other cardiac functions, such as stroke volume and cardiac output. You can see that when we pre-glycate the injected hydrogel, we do not get a protective effect in terms of reducing scar size that we see with the normal hydrogel. The normal hydrogel, again, as we've seen in the previous study, was able to recruit increased numbers of wound healing macrophages as determined by expression of CD206, whereas the pre-glycated hydrogel was not able to increase the number of these reparative cells. So as another next step in this project, since we've now identified that collagen is a target for glycation by methylglyoxal derived in the heart after MI, we are now seeking to increase the capacity of the material to absorb methylglyoxal. So we know that 10% of the mass of natural collagen consists of methylglyoxal trapping amino acids. So instead of using the full collagen molecule, as we have done in the past, David, a postdoctoral fellow in the lab, is now developing peptide-based hydrogels using collagen-like peptides that mimic the structure of collagen. And by using these peptide hydrogels, we're able to increase the number of target sites for methylglyoxal. And so we can make injectable hydrogels that will have greater than 10% trapping capacity. And so that is what he's working on now. We're going to develop materials with different percentages of arginine and lysine residues, which are the main amino acid targets for these materials. And we are going to then look if we can get a dose effect in terms of their ability to trap methylglyoxal. We'll be combining these peptide materials with fisetin, which is a flavonoid that we've previously reported can improve myocardial infarction after its delivery. So this is a summary of the published work conducted by Chala, a PhD student in the lab, whereby fisetin was injected along with collagen hydrogel into the myocardium at 3 hours post-MI. And it was able to reduce oxidative stress, inhibit inflammation and apoptosis, reverse or prevent some of the adverse cardiac remodeling, and improve cardiac function. And so now we're going to be combining that strategy with the peptide materials so that we have a system in which methylglyoxal can be sequestered away from the myocardial tissue in the injected material, and the fisetin will be released from the material into the environment to be absorbed by the cells and further provide protective effects against methylglyoxal. So another strategy that we're using to minimize the effects of methylglyoxal post-MI is the use of GLO1 loaded nanoparticles. So we're loading nanoparticles with the glyoxylase 1 enzyme and delivering it systemically. That's to then target the injured myocardium. And this is work that was started by Chala, and Gigi is now continuing the project in the lab. And we've done this in collaboration with a group in China. They're the ones who have developed the nanoparticles and have kindly shared them with us for cardiovascular use. So I'll just briefly explain the nanoparticle itself. So it is based on hyaluronic acid, which is a natural ligand for CD44. And CD44 is a receptor, a cell surface receptor that is upregulated in endothelial cells after myocardial injury. And so we'll be able to use these particles to target the increased CD44 expression in the heart after myocardial infarction. So another feature of these nanoparticles is that they are fluorescent green, so we're able to visualize them. We inject them by tail vein systemically into mice after MI, and then they will, through the circulation home to the heart, get bound to the CD44 receptors in the myocardial tissue and get taken up inside the cell. And once they're inside the cell, then they will release their contents, thus delivering the glyoxylase 1 to the cells in the injured tissue. So we performed in vitro studies to look at the uptake of the nanoparticles and the release inside the cells. So we cultured endothelial cells, treated them with methylglyoxal, and then added our nanoparticles. As you can see, these under normal conditions, endothelial cells have a lower level of CD44 expression, but upon exposure to methylglyoxal, the CD44 expression is greatly increased. And consistent with this increased expression, we see increased uptake of the fluorescent green nanoparticles when the cells express more CD44. And in this study here, we used a blocker, so CD44 blocker, to block the receptor. And you can see that when we do this, we have reduction in the uptake of the nanoparticles, demonstrating that the nanoparticles are quite specific for binding to the CD44 receptor for uptake. And here we measured the glyoxylase 1 expression inside the cells after treatment with empty nanoparticles or GLO1 loaded nanoparticles. So here in this condition, we did not treat the cells with methylglyoxal, and you can see there's no difference in the glyoxylase 1 expression between cells treated with nanoparticles that are empty or containing glyoxylase 1. However, once we treat the cells with methylglyoxal, we see an increase in GLO1 levels in the cells that are treated with the GLO1 nanoparticles. So we are able to deliver the enzyme to the cells. So in vivo, we again used a mouse model, myocardial infarction. Mice underwent surgery to induce infarction, and then 3 hours post-MI, we delivered the nanoparticles via tail vein injection, and then followed these animals for out to 4 weeks, looking at echocardiography, using echocardiography for cardiac function analysis and collected tissues as well. So here are some results of the bioluminescence imaging experiments we performed to look at the accumulation of our injected nanoparticles in the heart. So here at 3 hours, you can see that nanoparticles, which fluoresce green, are detected by the bioluminescence imaging to be accumulating in the myocardium, both empty nanoparticles and GLO1 loaded nanoparticles are being taken up equally. And then at 24 hours, we still see that they are being taken up inside the heart to a lesser degree than at 3 hours, but over 24 hours, nanoparticles continue to home to the heart and get retained there. In terms of the glyoxylase 1 expression, you can see that GLO1 nanoparticles are able to increase the glyoxylase 1 expression in the heart 3 hours after they've been delivered, compared to the empty nanoparticles and PBS-treated mice. In addition, we looked at the oxidative stress levels and observed reduced oxidative stress in the animals that were treated with GLO1 nanoparticles compared to empty nanoparticles and PBS, both at 3 hours and at 28 days post-delivery. In terms of cardiac function, the delivery of GLO1 nanoparticles was able to increase ejection fraction 28 days post-MI. In addition, we saw improved cardiac contractility in the GLO1 nanoparticle treated mice. GLO1 nanoparticles also reduced the scar size and reduced cell death post-MI compared to the other two treatment groups. Capsule exposure leads to increased CD44 expression in endothelial cells. Hyaluronic acid-based nanoparticles can target the increased CD44 receptor expression and deliver GLO1 into the cells. Systemic administration of GLO1 nanoparticles to mice with MI resulted in smaller scar size and improved cardiac function. Therefore, targeted delivery of GLO1 to the infarcted myocardium may be a promising therapeutic approach to reduce the negative effects of methylglyoxal post-MI. So where are we going from here? So unfortunately, the precursors that we were using to make our nanoparticles are no longer available worldwide. And so we now had to redesign the nanoparticles. So again, with our collaborators in China, they've sent us new formulation that we are now using in the lab and having to retest and reconfirm their properties. So these are now ready to begin in vitro testing in the next few months. So we're hoping to get back to where we were relatively soon. In addition to the permanent ligation model that we've been using in the lab, with this project, we'll also be looking at using a myocardial ischemia perfusion model for testing, which we are doing in collaboration with Tammy Martino at the University of Guelph. And we'll also then look at the effect of our treatment on the inflammasome, which is something that we've not previously looked at. So since the nanoparticles that we're delivering are targeting CD44, which is on endothelial cells, but we're also seeing beneficial effects on the cardiomyocytes. So we want to better understand how is preservation of endothelial cells resulting in improved cardiomyocyte function, survival, and function. And to do this, we are starting with some in vitro culture experiments, which is being done by Ramis, a PhD student in the lab. So he's using a co-culture system where the cells can be separated and cultured independently. So we can treat cardiomyocytes in one side of the culture with methylglyoxal, and then leave the endothelial cells untouched, mimicking their protection from the GLO1 nanoparticles. you can culture them this way for a period of time and then add additional media. And now the cells are able to communicate with each other. So we can see what healthy endothelial cells are able to do in terms of protecting cardiomyocytes. So far we've looked at viability and observed that the co-culture of the endothelial cells and cardiomyocytes is able to improve cell survival when the cardiomyocytes are exposed to methylglyoxal compared to when they are not able to communicate with endothelial cells. So this is cardiomyocytes on there. And down the road, then once our nanoparticles are ready to be tested, then we will also be testing their therapeutic efficacy in a similar co-culture system. So now I'd like to move on to the project where we're investigating the effect of exercise on methylglyoxal post-MI. This is work being led by a postdoctoral fellow in the lab INIS. So it's already been shown that exercise can confer benefits in a non-pharmacological way through increasing cardiac output, increased blood flow through the coronary arteries. It has a positive effect on maximum oxygen uptake, and it can also reduce blood pressure. So the aim of this project is to examine the effects of exercise on methylglyoxal-derived ages in patients with MI. This work is being done in collaboration with Jennifer Reed. So she's already conducted a study in which patients underwent different forms of exercise as rehabilitation after MI. So one group underwent a program of Nordic walking. Another performed moderate to vigorous intensity continuous exercise, or MICE. And then another underwent high intensity interval exercise training, or HIIT exercise training. And they performed these programs for a period of 12 weeks. Blood was collected before engaging in the exercise and after the 12 weeks were completed. We also obtained blood from a group of healthy individuals for comparison, and we analyzed the levels of MGH1, which is again the primary age formed by methylglyoxal, and we also looked at some of the secondary age products, CML and CEL. We looked at these both in the serum and plasma through ELISA, as well as mass spectrometry. So here are some of the results in female patients. As you can see, compared to healthy individuals, the levels of MGH1 in the serum and plasma were increased in the MI patient group. We also observed increased levels of CML in the MI group compared to healthy individuals in the serum only. And we saw increased CEL levels in patients with MI in both serum and plasma. So then looking at the results of exercise compared to the baseline levels of these three products, you can see that the HIIT exercise program was the only one that was able to reduce MGH1 levels compared to before the exercise was started, both in serum and plasma. And there were no effects of exercise seen on the other two products that we analyzed. Therefore, it appears that the HIIT exercise program is beneficial in terms of reducing circulating levels of MGH1. And because of this, we're using an exercise model in mice for the HIIT program to better understand how the exercise is reducing MGH1 levels in the circulation. So we developed a program, treadmill running program, similar to what the patients undergo clinically. And this was done with the help of Han Kim to set up the exercise treadmill machine. So in the mice, we observed that the HIT training program was able to increase cardiac function in terms of ejection fraction, both in male and female mice. Scar size was reduced in the male mice that underwent the HIT training program, but this was not statistically significant in the female cohort. In terms of cardiac contractility, again, in the male group that underwent the exercise program, we saw improved function, but this was not statistically significant in the female group, although more animals are still undergoing the protocol, so it may just be a sample size issue at this point. There is a trend, similar trend in the female group. So in summary, circulating levels of MGHs are increased in patients with MI. Exercise training can lead to decreased expression of MGH1. And so the ability of exercise training to reduce methylglyoxal and its AGEs may contribute to patient recovery post-MI, and understanding the mechanisms of this phenomenon may help to improve and or customize treatments for patients. So these are the ongoing or future studies looking at the exercise effects on methylglyoxal. So in the mouse model, we will be measuring the circulating and tissue levels of the MGHs. We're going to examine the effect of exercise on the expression of glyoxylase 1 and its activity. And we're going to investigate potential pathways linking exercise with the regulation of glyoxylase 1 enzyme levels and activity. In the human patients, we will be comparing circulating levels of MGHs between males and females before and after the different exercise programs. And we're also going to be using mass spectrometry analysis for improved accuracy and higher throughput of the patient blood samples. And so we've got shown here some preliminary mass spec results demonstrating that the levels of MGH1 in males post-MI are higher than in females in the serum, but no difference was detected in the plasma. So the final project I'd like to present today is the work being done on the brain-heart connection. So we know that methylglyoxal is being produced and released by the infarcted heart, and the methylglyoxal products are circulating in the blood, and that these are increased in patients with MI. So MG and its AGEs are known to be able to cross the blood-brain barrier. There's not a lot of publications out there looking at that, but there are a couple showing that they can cross the blood-brain barrier. But it's unknown if MG and MGHs accumulated in the brain post-MI. So this is what Ramas has set out to do in the last year, is to look at whether some of these circulating AGEs post-MI are getting into the brain. And so by Western blot, you can see here he's got healthy brains from mice and brains from mice 2 days post-MI, looking at MGH1 and also the expression of glyoxylase 1. And you can see that even just 2 days after MI, there is increased levels of MGH1 in the brain of these mice. In addition, there's increased expression of the enzyme glyoxylase 1 in the brains of mice after 2 days after MI. So likely this is in response to the increased MGHs, and so glyoxylase 1 as the glyoxylase system as a defense mechanism against MG, methylglyoxal and its AGEs also is increased to try to deal with this accumulation. So that work that I just showed you was in females. We are also going to be comparing males versus females in the future work and looking at different brain regions to try to identify if there's specific sites within the brain that are targeted more than others. And so I've got some preliminary data here looking at this comparison of males and females in the hippocampus. So here, this is at 6 hours post-MI and at 7 days post-MI. looking at the levels of MGH1, so again, the dominant age product of methylglyoxal, you can see that there is a significantly greater level of MGH1 in the males compared to females at 7 days post-MI. And these are both higher at 7 days post-MI than in the healthy brain. In terms of glyoxylase 1 expression, we see greater expression of this enzyme in females at all 3 time points. So even in healthy females, there is a higher basal level of glyoxylase 1, and this is the higher expression is maintained after MI and continues to go up. Whereas in males, it appears that there is no change in glyoxylase 1 expression post-MI. So with that, I'd like to conclude my presentation, and I'm happy to answer any questions.
Very exciting and provocative data, both in terms of the nanoparticle aspect as well as the new brain heart connection, which is fascinating. But I think it's consistent with some of the biomarker work that we're doing. Some of it is very stuff, and this is fantastic. I don't know if there are any questions from the room.
Go ahead, Peter. I'll see you.
So Mark is going to ask the tough question. My question actually, I have so many, sort of things that come. Yeah, apologies. Yeah. I was I'm fascinated by the nanoparticle work, because mainly because the fact that, hey, it is possible then to utilize it, for example, in reperfusion therapy, right? Because this is the bulk of the patients, with acute myocardial infarction. So the delivery is really exciting from that point of view. So a couple questions. One is that does the nanoparticle, I think, you're kind of implying it in terms of the myocyte versus, sort of apart from the endothelial cells, where the receptor is located, does actually also affect the myocyte and, of course, the macrophages, which are really exciting in terms of repair. And could this be possibly combined with the other strategies, because it appears the effect is relatively, brief in the acute setting. For example, I'm making this up, like an mRNA for NRF2 or something like that, which actually gives a more sustained, sort of benefit over time. I'm just kind of free associating here. Yeah.
Yeah, for sure. The nanoparticles are probably able to deliver multiple different types of treatments. So yes, RNAs would be it'd be possible to deliver those through this mechanism as well. Another option is to deliver the particles at different time points post-MI. I mean, it is just injected systemically, so it's easy to do, provided that CD44 expression is elevated over the period of time that you would be delivering, which is something that we're looking at too, to see how long is CD44 elevated expression maintained post-MI. So we have a window that we would be able to consider for delivering therapy over time. And I forgot to mention it on my slide, I showed her picture. Jessica in the lab is working on another nanoparticle project where the nanoparticles will trap the methylglyoxal and its ages in the circulation, thus hoping to prevent them from getting into the brain. So that's another application of the nanoparticles as well.
Eric, a very nice presentation. As always, so clear, so well organized, and really a delight to hear the whole story about methylglyoxal. Going to be during some of our meetings, I always ask you stupid questions. What's the clinical applicability of this? So first one, to follow up on Peter's questions, nanoparticles. Can we use that in humans? Like, are there applications that we know, or it's an absolute no-no?
There there's no reason that they shouldn't be able to be used as long as the products they're made from are not toxic. And
so, you know, treatment of STEMI is you bring the patient quickly to the cath lab to reopen the artery, but even then, there's always myonecrosis going on. So you could easily envision if you had something that can give GLO-1 to a patient as they come, hopefully within a few hours of their first chest pain symptom, you could give them that. So what happens, the GLO-1 on a nanoparticle, what's the half-life of that? What kind of a pharmacodynamics is involved? What if it goes in people's skeletal muscle? Like, does it have a toxic effect? Do we even understand what the potential therapeutic role is?
I've not seen any evidence of a negative effect of overexpressing glyoxylase 1. I've seen the opposite, where in mice that throughout their born overexpressing glyoxylase 1 live longer. But I have not seen a negative effect of having too much glyoxylase 1 expression. All it would be doing, I think, is removing more methylglyoxal, so you're removing more toxicity. So I don't think there's a danger of getting glyoxylase 1 expression in other tissues.
Two very quick. How many groups are working on this, Eric? The methylglyoxal, is there like a huge number of groups, or are you pretty much alone?
Of groups that I've seen with publications anyway.
Last question.
And most of the ones I've seen are not working on it therapeutically. It's just the biology behind it.
So MGH1, you looked at the effects of exercise with Jennifer. That's a nice translational collaboration. That being said, you know, it's 12 weeks after. You think that all the damage has been done by them, right? It probably has been. So what if we take one of us here in this room and say, you know, we've been sedentary for a week, measure MGH1, and then Jennifer puts us on a treadmill 4 times a week for a week and measure it again. Is there going to be a difference? Do we have that kind of physiologic data? We all have circulating levels, presumably. Yeah. And do they go down with exercise? Thank you.
Yeah, I don't have an answer to that. I haven't seen a study looking at it in healthy individuals. But I would assume it probably would go down. That if you are sedentary and then you begin exercising, you probably lower your circulating levels.
That hits you pretty hard.
Yeah, everybody always function.
It's I have these with Baba music, you know, it's really actually very intense.
Eric, really nice presentation. Since everybody's talking about the nanoparticles, in your model, you're working in myocardial infarctions. I assume you fully ligate the descending aorta. Sorry, the descending coronary artery. So if you're injecting by the tail your nanoparticles, how are they getting into the infarct area? That's a curiosity question.
Yeah, I don't think they're getting into the infarct itself. They're getting into the border area and remote myocardium and preserving that tissue, preventing expansion of the damage, which would occur over time. Yeah, so I think the benefits in terms of like even looking at the myocardial contractility, where we saw it is in the border area that we have improved contractility, not in the infarct area. So it appears that the effects are not in the scar.
But in clinical application, because now reperfusion therapy is done so effectively, there's a general, you know, population reduction in infarct size already. You know, it's of course, you know, accompanied by improved survival. And so I do think that you know, in that type of setting, then, you know, going through the coronary arteries, you know, through the catheter is very, very realistic, you know, and help to actually distribute. So to follow up on that discussion, a couple of things, you know, well, I'm very happy that you're actually working together with Dr. Tammy Martino, who was actually my PhD student a long time ago. You know, she's doing amazing. So this is a great collaboration. The other two things to consider, you know, follow up on Mark's question, you know, so in terms of the nanoparticles, you know, so I know Darryl is working on exosomes, you know, so which could be one thing. And then we're working on lipid nanoparticles, you know, which has been, you know, already used for vaccines and things like that. And we're working on versions that can actually target the heart, you know, so I don't know whether, you know, these other packaging materials could also be used, you know, because they are certainly easier to potentially get approval, you know, from FDA, things like that. Yeah.
Mm-hmm. Yeah, and there may be ways of more specifically targeting them to the heart using potentially some of the same strategies.
Yeah, exactly. Yeah, so I think they're following the same principles, right? Yeah, and so maybe other questions. Maybe the last question I have is the I guess the potential application, you know, for this is in the acute setting, right? But you know, redox activation is really very, very, you know, sort of common in many other conditions, you know, whether it's diabetes, whether it's you know, sort of certainly heart failure and things like that. And has there been any you know, sort of application of this in addition to acute you know, sort of MI setting?
Yeah. We have not been looking at that, and as I mentioned, I haven't seen other groups working on therapies targeting methylglyoxal, at least not in the way that we are. So I don't think that's anyone's looked at that yet. I should also mention, I mean, we've designed the particles to target the endothelium because they express the CD44, which is a target for the hyaluronic acid. But circulating monocytes also express that receptor. So it's quite likely that the particles, since we're injecting them into the blood, are being taken up inside the inflammatory cells as well that are being recruited to the heart. So that's likely contributing to the effects that we're seeing.
Yeah, that's why my suspicion that the macrophages, you know, sort of were also being a positive, you know, benefit. So this is great. You know, thank you so much, Eric. Great work, very innovative, and you know, sort of getting to the exciting translational stage as well. You know, so looking forward to further developments in this whole area, you know, which is actually fantastic. And so I don't see any you know, specific questions online. So, but thank you again, and thanks everybody for joining us. And so there is no scheduled presentation next week because it's a long weekend, you know, but we'll continue the session afterwards. Yeah, so thank you so much again. Take care.